Good afternoon. I am Alexandria
And I am Athira Gopakumar.
We are going to be reviewing the article Venomous Extract Protein Profile of Brazilian Tarantula Grammostola Searching for Potential Biotechnological Applications by Marcia H. Borges and her colleagues. The article was published in 2016 in the Journal of Proteomics, which has an impact factor of 3.509, and the article itself has been cited 12 times. For our presentation, we took an excursion to the Australian Reptile Park, hoping to see the Grammostola tarantula. But unfortunately, there were none. However, there was this lovely Mexican red-kneed tarantula, as shown here, that shows you how terrifyingly big they are. The Grammostola tarantula is a hunting spider found in tropical regions around the world, and paralyze their prey by injecting them with venom from their chelicerae. But why study the venom of these spiders? Venoms are sources of toxins containing molecules that are of interest for the medical industry, due to their ability to bind to specific cell targets and modify their functions. This article's purpose was to study and characterize the Grammostola venom components and its molecular, physiological, and pharmacological diversity, to discover components with potentials for searching for new active molecules. This study uses an analysis method called mud pit to study the venom. However, previously, this approach was not used to analyze toxins. Here are 4 types of approaches mentioned in 4 of the articles cited by this study. This is an overview of the method performed in this study, which was provided by the article. I will briefly explain the extraction of the venom from the tarantula, whilst Athira will explain the rest of the method. "13 specimens in total, male and female, underwent electrical stimulation of the chelicerae to release the venom, which were then collected and solubilized in ice-cold acidified water and centrifuged to remove the insoluble materials. The collected supernatant was assayed and protein-extracted aliquots were separated into single-use samples and lyophilized. This video that was captured by myself at the Australian Reptile Park is the extraction method for venom for anti-venom purposes, but was not for this case.
The current approach for prospection of venom toxins is through proteomic methods, which helps in identifying larger molecules in the venom. Multidimensional protein identification technique, mudpit, was applied to understand the venomous extract of G.iheringi spider. This method helps us understand the physiological and pharmacological properties as well as the biotechnological potential it possesses. Mudpit is a chromatographic proteomic technique in which peptide mixtures are prepared from a protein sample and is directly loaded onto a triphasic microcapillary column, which is packed with reverse phase strong cation exchange and reverse phase HPLC. After the peptide mixture is added into the triphasic microcapillary column, it is placed in line with a tandem mass spectrometer. The data generated from the tandem mass spectrometry from a mudpit run is searched using softwares to determine the protein. The sample venomous extract preparation was performed as follows. 100 micrograms of the sample was solubilized in 100 microliters of water and precipitated with trichloroacetic acid, and a final concentration was obtained of 25% and held overnight at 4 degrees centigrade. The samples were centrifuged for about 30 minutes at 4 degrees centigrade. The pellet was then washed twice with 500 microliter of ice-cold acetone and then air-dried. The pellet was then solubilized in 100 microliters of 8 moles urea in 100 millimoles of tris HCl with a pH of 8.5. The disulfide-bridge cysteine residues were reduced by 1 mole of the tris phosphine to a final concentration of 5 millimoles and incubated at room temperature for 20 minutes. The thiol groups were alkylated by adding 500 millimoles of iodoacetamide to a final concentration of 10 millimoles, and the solution after which was incubated at room temperature for 30 minutes in the dark. Trypsin was added to a ratio of 1:50 and incubated at 37 degrees centigrade overnight in the dark. The digestion was stopped by adding 90% formic acid to a final concentration of 5%, followed by centrifugation for 30 minutes at 4 degrees centigrade. The supernatant was collected and stored at -20 degrees centigrade. The mud pit analysis. The digested peptide samples were loaded onto a capillary column packed with 2 different matrices, being a strong cation exchange followed through by reverse phase. Then, peptide desalting was performed using a 12 cm column with the same reverse phase matrix. Mass spectrometry was performed utilizing an automated 12-step mud pit separation method. Peptides eluded from the mud pit column were directly electrosprayed into the LTQ XL Orbitrap Max spectrometer. High-energy collisional dissociation fragmentation was employed, and MS1 data was acquired at a resolution of 60,000. Various softwares were used to identify sequences and process the data that were obtained. The main softwares used were PEP Explorer, Blast2Go, and InterProScan, and Gene Ontology.
Using PEP Explorer, 886 protein sequences were identified and simplified to 395 sequences. Of these 395 sequences, 30% matched previously described proteins, and 6% were unknown. 63 toxins with a coverage of 90% were discovered. Most were similar to toxins expressed by other spiders and scorpions, 24 had unknown targets, and 8 were neurotoxins similar to those described previously in Grammostola spider venom. This showed that mud pit analysis was able to sequence and identify more molecules than were sequenced and identified by previous methods that had been mentioned in this presentation. This article demonstrated the potential of broad-spectrum characterization of the composition of animal venoms through mud pit shotgun genomics to identify and sequence great amounts of proteins in venom samples than methods previously used. The mud pit shotgun analysis identified over 300 proteins in the extract, including some that have never been identified at their protein level, and some that have evidence at a genomic level only. Therefore, we highly recommend the mud pit genomic analysis as a sensitive and satisfactory application to obtain information about animal venoms at a broad spectrum level. The next few slides will just show our references. Thank you for listening.